All right, thanks for offering me this opportunity to present my work. My name is Shi. I'm a grad student working in the laboratory of Professor Bogdan Pasternak. Today, I'll be talking about our recent methods to estimate local genetic correlation from GWAS summary level data and how we use this estimator to localize shared genetics between complex traits. But first, I'm going to briefly talk about genome-wide association studies. So in a typical large-scale GWAS, you first collect tens of thousands of individuals, you group them by case and controls. You then go ahead to genotype these individuals using some genotyping technology. After that, you scan through the genome to look for association between each trait and the SNP. The result is often visualized as a scatter plot, where the x-axis is the location on the genome and the y-axis is the strength of association. This scatter plot is often called the Manhattan plot. So over the past decades or so, thousands of GWAS have been performed, which identify tens of thousands of trait-associated variants. These studies have also created a huge resource of GWAS summary level data that are publicly available, which really opens up a lot of opportunities for geneticists to study the genetic architecture of complex traits. So if you look at the Manhattan plot for some of the traits, you will notice that some of them have very strikingly similar pattern of association signals. As an example, for the pair of traits, low-density lipoprotein and triglyceride, they have very similar Manhattan plots. As you can see, all the peaks are shared for these two traits. So this is an indication of shared genetic architecture for these two traits. For example, the pair of traits LDL and triglyceride could be both affected by a pathway, which is in turn affected by the same set of SNPs. So a widely used approach to quantify the degree of genetic sharing is genetic correlation. So in genetics, we often model a trait as a summation of its genetic effect and its environmental effect. And then the genetic correlation, for example, between the trait LDL and triglyceride is then the correlation between the genetic components of the two traits. So previous method called cross-trait autoregression can estimate the genome-wide genetic correlation between a pair of traits from GWAS summary association data. And this method tells us that the genetic correlation between LDL and triglyceride is on the magnitude of 0.4, which means that if you know the genetic component of the LDL, you will be able to predict with a modest degree of accuracy the genetic component of triglyceride. However, as you can see, genetic correlation is a single number, and therefore you cannot use this number to identify loci that are shared across two traits. So to solve this problem, we propose to estimate local genetic correlation. So to define local genetic correlation, we first truncate the genome into m independent loci. And we model a trait as a summation of the genetic component at each locus on the genome. And therefore, the genetic correlation, for example, between a pair of traits LDL and triglyceride is then the correlation between the genetic component of these two traits at the ith locus. And effectively, local genetic correlation decomposes the genome-wide genetic correlation into each small region on the genome. And because we have a number for each locus on the genome, we can use this number to localize regions that are shared across two traits. So to illustrate the concept of local genetic correlation, let's look at a toy example involving two pairs of traits. Here, both pairs of traits have the same total genetic correlation. However, for the pair of traits in the left figure, the total genetic correlation is dominated by this large positive local genetic correlation and cancel out by a lot of negative but very small negative local genetic correlation. However, for the pair of traits in the right figure, the total genetic correlation is an aggregation of a lot of positive but all very small local genetic correlation. So the idea is that although two pairs of traits may have the same total genetic correlation, their local distributions might be very different from each other. So to estimate or to quantify local genetic correlation, we developed a method called p-HESS. So p-HESS takes as input two sets of GWAS summary data, one for each trait, as well as the LD matrix for the SNPs in the data sets. Importantly, the LD matrix does not necessarily have to be estimated from the GWAS samples themselves. Instead, it can be estimated from an external reference panel, such as the 1000 Genome Project. This really allows us to apply our estimator on any set of GWAS summary data that are publicly available. So for a pair of traits on each locus on the genome, p-HESS will output three numbers. So one is the local genetic correlation, covariance between the two traits, and the other two are the local SNP heritability for the two traits. So we went ahead and applied our estimator on 36 summary level data, which gave us a total of 600 pairs of traits, and we estimated the local genetic correlation on 1,700 loci. And we compared our estimates of total genetic correlation with those obtained by cross-trait statistical regression. And as you can see, our estimator is very consistent with cross-trait statistical regression, which reassures that our estimator is performing as we expect. So then we went ahead and looked for loci that are shared across two traits. So this figure here, so we first look at pairs of traits for which the genome-wide genetic correlation is significant. So the figure here is showing you the loci that we identified, as well as the genetic correlation, as well as the traits that are driven by this locus. So unsurprisingly, for pairs of traits with significant genome-wide genetic correlation, there are a lot of loci that contribute to this genetic correlation. So in total, we find that there are 21 loci that show significant local genetic correlation for 12 pairs of traits. And I'd like to highlight these two loci on chromosome 2 and chromosome 11, which show significant local genetic correlation for a lot of lipid traits, such as HDL and triglyceride. So we look for genes that are covered by this region. And we found that these two loci covers the gene APOA and the APOB gene, which is known to be associated with multiple lipid phenotypes. Surprisingly, a pair of traits does not necessarily have to have significant genome-wide genetic correlation for them to have significant local genetic correlation. This is possible because a pair of traits may have a lot of positive local genetic correlation, but they might also have a lot of negative local genetic correlation, which balance out the positive ones. So if you look at the total, the sum, you will see a number close to 0. But if you zoom in at each region on the genome, you'll still be able to see some signal. This is clearly true for the pair of traits HDL and LDL. As you can see, this pair of trait has a lot of positive local genetic correlation, as well as a lot of negative genetic correlation. But the sum is close to 0. However, if you look at each individual loci, you'll be able to see a strong local genetic correlation signal. So we applied our estimator. So we then look at all pairs of traits for which the total genetic correlation is close to 0. And surprisingly, we find that there are actually 7 loci that show significant local genetic correlation for 10 pairs of traits for which the genome-wide genetic correlation is close to 0. And I'd like to highlight this locus on chromosome 6, which shows significant local genetic correlation for lots of blood phenotypes, such as mean cell hemoglobin and red blood cell counts. And we went ahead and looked for genes covered by this region. And we found that this region covers the HBS1L gene, which is known to be associated with a lot of blood phenotypes. So our estimates of local genetic correlation can also be used to infer causality between pairs of traits. So for example, if we look at the pairs of traits, body mass index and triglyceride, we see that the local genetic correlation is positive across the entire genome. And this is actually an indication that there might exist some causation between the two pairs of between the two traits. However, just based on this figure, we cannot figure out what's the direction of causality. So to infer the direction, we first look at the causal loci for body mass index. And we see that the SNPs in the causal loci for body mass index have a consistent effect on the level of triglyceride. However, when we look at the causal loci for triglyceride, we see that the SNP in this loci does not have a consistent effect on body mass index. This is an indication that body mass index causally increases the level of triglyceride. So we went ahead and evaluate the local genetic correlation at causal loci for body mass index, as well as the local genetic correlation at the causal loci for triglyceride. And we see that the local genetic correlation at body mass index is much larger than the local genetic correlation at loci for triglyceride, which is consistent with the model that body mass index causally increases the level of triglyceride. And we know that this result is consistent with the result reached by previous work. So to summarize, in this talk, I presented our estimator p-HESS to estimate local genetic correlation from summary level data. We use this estimator to localize regions on genome that are shared across two traits. We also use this result to provide evidence of causation for pairs of traits. So in the end, I'd like to acknowledge our lab mates for their help during the course of this project. I'd like to also thank our collaborators at BYU and Harvard School of Public Health for their helpful discussion. And I'd like to end my talk here. I'm happy to take any questions you might have.